Thanks for coming, everyone. So I'm going to tell a story about, for me, a first foray into a clinical fly collaboration and how we've ended up finding out some interesting things from this, which hopefully also impacts patient outcomes. So I'll just introduce you very quickly to this very basic developmental model here. You can see this is a sort of schematic of neurogenesis. So you have a multipotent stem cell with self-renewing capacity, which can divide and also form intermediate progenitor cells. And these have limited mitotic potential and can divide further to create the neurons and the glial cells that comprise the nervous system. So we think a lot about how these cell states are specified during this developmental time course, um, with, of course, regards to differentiation. Um, but it's less, I think it's less well understood how these cells set up a cell state that is then maintained across that entire life course during development and also later. So by cell states, of course, I'm talking about differentiation. And so during the process of differentiation, what's quite well established is you have epigenetic processes that deposit marks that cause long-term silencing of genes that are not required for a particular cell state, or else continue to express the correct marker genes for that neuron. Um, but we also need to think about cell cycle exit, since these cells, neurons in particular, exit the cell cycle and then stay that way for their entire lifespan. So how are the mechanisms that initiate that cell cycle exit established and then maintained during the lifespan is a key question. So here's just a cell cycle diagram, which most people watching will be familiar with. And I just want to introduce this little mechanism which regulates these cell cycle genes. So this small co-activator complex, which in its simplest terms consists of an E2F protein and its binding partner DP, binds at the promoters of S-phase genes to cause their expression during the appropriate stages of the cell cycle. But during G1 phase, what you have is a transcriptional co-repressor, this retinoblastoma protein, or RB, which antagonizes the action of that E2F activator complex. And these cycle during the cell cycle, and it's thought that during cell cycle exit, this RB retinoblastoma protein will cause cell cycle exit to occur. So this project really started when some clinical collaborators of ours approached us with a cohort of patients who have mutations in one of these retinoblastoma genes, RBL2. And what's interesting to me about this is that retinoblastoma proteins are well known for causing tumor phenotypes, which is in the name retinoblastoma, so these childhood ocular tumors. But these patients, in fact, didn't have any sign of that. Instead, you have quite a broad clinical spectrum, which is characterized by things like intellectual disability, but also movement phenotypes, dystonia, ataxia, and also notably microcephaly. So, as I say, quite a broad spectrum of neurological phenotypes. So, our task initially was just to look in our fly models to see to what extent can we confirm that retinoblastoma proteins' involvement in these kinds of processes. So as I say, we want to provide experimental support for these genotype-phenotype linkages, since we have quite a small number of patients, that's useful to do. And then to identify the afflicted cell types and developmental stages to understand the pathology a bit better, and obviously then go on to understand the molecular mechanisms a little bit better. So I'll just whiz through some of the experiments we did quickly to confirm that what we are seeing in the fly is similar to the human patient. So what we've mainly focused on is quite a well-studied hypermorph allele, which causes reduced expression of RBF. But it was unclear whether these animals really had any neurological consequences. So just quickly, the patients have this quite significant developmental delay. And we saw a similar thing in the flies, taking the time to eclosion as a developmental landmark. It takes the flies longer to get there. I should mention everything's in an isogenized background here. Um, Jepsen Lab focuses really on movement disorders. So we have a whole battery of tests to look at this in the fly, but just highlighting here these Drosophila activity monitors in which fly moves freely and crosses an infrared beam that then's monitored over 24 hours, and also the climbing assay, and we can see decreases in movement in both of these assays. And these behavioral phenotypes extend even to more complex phenotypes, such as sleep. So we see reduced sleep in the night in both males and females of these hypermorph flies, as well as changes in the anticipation of the changing light regime. And also, finally, for morphological phenotypes, the humans have microcephaly, and we also see this in the fly. If we quantify the size of their brains, they're significantly smaller. So just moving on to look at this in a little bit more detail, we decided to look at the larval brain. So here's the Drosophila larval brain, and what we can see is that there are actually more apoptotic cells in the larval brain. And this is actually not completely unexpected. There's a nice paper from a few years ago that looks at RBF apoptosis in the developing eye, and they see a similar thing, increasing apoptosis in some of the precursor cells. So this is, we think, probably explains to some extent the microcephaly and some of the behavioral phenotypes. But what we also looked at was the adult brain and noticed that there actually wasn't any continuing apoptosis in the adult brain. But what was surprising to us is that when we look at the single cell data that's publicly available, what we can see is that RBF is actually extremely highly expressed in these clusters that also express neuronal markers, in this case NSIB. So we're looking at the red in the diagram here. And when we chase this up with our own reporter line, we can see that this is indeed the case. RBF is quite highly expressed in neurons. So this was surprising to us because we had anticipated that RBF is required for cell cycle exit, but we wondered what it's doing continuing into the adult brain where there is no ongoing neurogenesis in the fly. So these are all mature neurons at this point. So what we found is that when we knock down RBF with NSIB-Gal4, a neuron-specific driver, which is expressed actually fairly late in the differentiation of neurons once they're already forming synapses, we can see that there's a significant loss of activity. And what was perhaps even more surprising is that if we use a US RBF to put RBF back into the hypermorph background, we get a significant rescue of some of the behavioral phenotypes. So whilst apoptosis in the larval brain is clearly underlying some of the phenotypes we see, most notably probably the decreased brain size, and maybe some of the behavioral phenotypes, there's also this component of RBF activity ongoing in the adult neurons. And to check this even further, we used a conditional knockdown just in the adult animals and saw the same thing. We could, when we knock down in the adult neurons, we see a loss of decreased activity. And when we add RBF back, we're able to rescue some of this phenotype. So we wondered what was going on here. There's been several reports in different tissues that say that RBF is involved in perhaps the differentiation of cell type specific genes or the regulation of metabolism. But when we did RNA-seq on these adult heads, what we saw was basically that RBF doesn't really appear to be regulating any non-canonical genes all that much. And it's specifically these DNA replication cell cycle genes that are misexpressed. So you can see in the volcano plot here, there's lots of upregulation of genes, so moving towards the right of the plot. And the Go analysis confirms that these are genes that are primarily involved in DNA replication. So at this point, I did a bit of data mining because I was curious about what RBF is doing in the adult brain. And I could see that actually in adult neurons, chromatin accessibility remains extremely high. So it's clearly RBF is not inducing an epigenetic state at cell cycle exit or contributing to that. And in fact, looks like it's likely required for ongoing silencing. And this data hot off the sequencer, so sorry it doesn't look so nice yet, but you can see that RBF remains bound at the promoters of these DNA replication target genes. And this is also the case. Sorry, I skimmed over the ATAC data. In human neurons, you can also see that this is the same. So this is a conserved feature. Um, so we now understand a little bit better what's happening in terms of the expression of these genes. Um, but what's actually happening at the cell level? Um, so what we see is that, in fact, when we take some of these brains out and just look at them under the microscope, what we can see is that a lot of these neurons are actually increasing in size, and sometimes by orders of magnitude. Um, so what we think is happening here is that instead of re-entering a canonical cell cycle and increasing in cell number, these cells are actually becoming polyploid through endocycling. So the expression of cell cycle genes in this context is sufficient to drive increased chromosome content. I'll stress that this is quite preliminary, but my preliminary quantification of DNA content in these cells also reflects this. We have more DAPI signal intensity in these cells. So to conclude, we find that RB proteins are required for normal neurological function in humans and in flies. We find that RBF-linked phenotypes arise from cell cycle perturbations and cell death, which may be predictable in the developmental context. But in postmitotic neurons, there's an ongoing function for RBF which doesn't cause apoptosis. And that RBF continues to regulate cell cycle gene expression independent of an epigenetic state in mature postmitotic neurons, and that loss of RBF causes re-entry into the cell cycle. So just to say thanks to the Jepson lab and everyone involved, and the clinical team who did the RBL2 patient study, some of whom are listed there. And pleased to say that some of this work was accepted for publication recently, and you can scan that square to see some of the data. So I think I've whizzed through that a bit, but if there are any questions, I'll be very happy to take them.
Wonderful. Thank you very much, Gabriel. I think that's a great example of finding a surprising result and following it up. So yes, please use the Q&A box to ask any questions. In the meantime, I wondered if you could comment on whether you think this mechanism is specific to neurons or whether similar principles might be applied to other cell types. Do the patients have any other phenotypes outside of the brain?
Yeah, it's interesting. I would have expected them to, but actually, it seems like it's predominantly neurological, and it could be that this is because RBL2 looks like it's the predominant RBL isoform in the human brain. There is, of course, the canonical RB gene and also RBL1, which may be performing a similar function in different tissues.
Okay. So we
But
have a
yeah,
question.
there could be other phenotypes that we haven't seen.
Sure. Thank you very much. So we have a question from Samil who's asking, have you tried removing the E2Fs in your RBF neurons to see if they rescue the phenotype?
Yeah, so that's something that people have done in other studies and quite consistently see that E2F1 knockdown is sufficient to rescue RBF, as you suggest. But I haven't been able to rescue as yet. And I think that I have some preliminary data which seems to suggest that E2F1 and E2F2 might be doing something slightly different in neurons to what they're doing maybe in other tissues. So that's something that I'm working on, but yeah, haven't been able to get a full rescue yet, but maybe a partial rescue. So it's complicated.
And sort of out of curiosity, is there a gain of function mutation in RBF, and what's the phenotype there?
There is a mutation, a fly allele, which is it has a constitutive, it's a phosphorylation site mutant, which is constitutively active. And again, very preliminary data. I haven't had the time to do this experiment properly yet, but it seems to not have any overt neurological phenotypes, but is perhaps less long-lived.
Okay. And Any other questions? If not, I can end on a question. So the cell cycle is, fundamental to development. So how do you kind of think that variations in the cell cycle and the phenotype we're seeing have been accounted for during evolution? Do you think that would you have expected more mechanisms to protect the cell cycle than you're seeing at the moment?
Um, yeah, I think the really interesting thing about this, which I didn't really touch on, is that there actually is some data to suggest that in neurons particularly, uh, re-entry into the cell cycle might be implicated in a neuroprotective role.
Okay.
So there's a lovely paper from Laura Petita's lab that showed that polyploid neurons increase as a function of age,
Hmm.
and also may be protective against DNA damage. So my hypothesis at the moment, taking all of that data into account, is that RBF is sort of acting as a switch to be able to quickly allow for cells to re-enter the cell cycle. Obviously, I've shown that that is not necessarily beneficial to the organism if it happens en masse, but
Hmm.
perhaps there's a trade-off that happens ensuring cell survival, perhaps with a concomitant reduction in neuronal function or fidelity.